Yeah. All right. So we are discussing some techniques regarding the analysis of action potentials, and we are now looking at correlations in particular. So quickly go through what we have done. We looked at random variables and the probability measure, sigma field, etc. That's used to define a random variable. We saw that when a neuron fires randomly, then the spike counts follow a Poisson distribution. And that's what we checked in our data, whether neurons are indeed firing in a Poisson-like manner. And it appears that they do. This is this slide here, where the fits of the Poisson distribution match the data very well. We also checked whether the inter-arrival times of spikes follow an exponential distribution, and that seemed to be the case as well. In the previous class, we looked at correlations. We defined what correlations are and why they are important in neural recordings, or how can they influence the way neurons process information about the external world. In our data, we checked whether firing of neurons of the two neurons that we have, is it correlated or is it independent? We looked at their definition. What does it mean for something to be independent and correlated? And we looked at the joint density function. This is this function here. And what should its shape be when if the random variables are independent? So this is how it should have looked like. Independence means that you're just multiplying one marginal distribution, which is what you have here, with one of these values. So it has the same shape as along any row as well as in any column. Right? And then this is statistical testing that needs to be done to know whether these are different or not. The statistical tests I have not discussed, not something we'll cover in this class, but this is how we check for independence. And correlations, again, based on this formula here. So that's what we did in the previous class. Okay, so now we are going to build on this and talk about auto and cross correlation functions. And that will be the end of this part. Okay, so now let's talk about auto and cross correlations. So again, as before, let's start by asking a question. In neuroscience, there's a lot of sort of importance that has been given to the fact that if multiple neurons at a given brain area fire together in a synchronous fashion, then the neuron that all these other neurons are connected to, that neuron can easily respond, because all the neurotransmitters that are released by the previous neurons, those neurotransmitters will come at the same time and therefore cause a larger depolarization and therefore generate an action potential. Okay, so if two neurons N1 and N2 are connected to the same downstream neuron N3, so right now N1 and N2 are my two neurons in V1, and let's say they are connected to this N3, which is in V2, which is the next area that V1 neurons project to. We don't have data from N3, but we do have N1 and N2. So it's easier to excite N3 if N1 and N2 fire synchronously. This actually is a profound theory called binding by synchrony, which was proposed several decades ago, and it's still something that people are very excited about. So the question is that in the data set that we have, are neurons firing synchronously? Now, there is actually some evidence that when the monkey pays attention to to a particular stimulus. So this is one case where the stimulus onset is at 1 second. The red and the green dots correspond to the action potentials fired by two different neurons that are being recorded simultaneously. And the blue dots indicate the times when both the neurons are firing together. So these are the synchronous events. Okay, and this is the case when the monkey is attending to the stimulus. This is the case when the same stimulus is presented, but the monkey is not paying attention to it. And as you can see that primarily there's some increase in firing rate as well. But the main thing that is happening is that the two neurons tend to be firing more synchronously when the monkey pays attention to the stimulus. Okay, so somehow attention increases the synchrony between neurons. So this is an idea that has been around for a while. So we want to test in our data whether there is any evidence of synchrony.
So,
okay, yeah.
in the previous slide, there was a line that said oscillations were not observed. There was no evidence.
Yes, that's right. So let me tell you what it means then. Although this paper found no evidence of oscillation, so let me tell you what I mean by this line here. So when this binding by synchrony hypothesis was proposed, two neurons can be firing completely randomly in the sense that their individual firing can be Poisson-like, but they still can be completely correlated with each other. Correlated, completely synchronous with each other. So suppose there's one neuron that's firing in a Poisson-like manner and a second neuron that perfectly follows the first neuron. So both their firings, when you look at them individually, are Poisson-like. But every spike is synchronous, right? So the raster plot that we have seen in the past, the second one could be a copy of the first. So both of them are completely random, but they are synchronous nonetheless. However, this binding by synchrony hypothesis, when it first came about, it was based on this very cool observation that neural assemblies, the neurons that fire, not only fire together, they also oscillate at a particular frequency, which happens to be this about 40 hertz or so. It's called the gamma rhythm, which means that these neurons fire together, but also fire every 25 milliseconds, preferably. Okay, so there's a spike that comes, and then after 25 milliseconds, there's another spike that will come. Then after 25 milliseconds, there's another spike, and so on. So it's synchronous as well as rhythmic. Okay, and this binding by synchrony hypothesis was proposed based on this synchronicity as well as rhythmicity. So when I wrote this, just wanted to say that here there's evidence of synchronicity, but no rhythmicity. Okay.
Mm-hmm.
But when we ask our question, of course, we will ask both. So in the neural data that I have given you, I have given you data from two neurons. I want to ask if there's any evidence of synchrony between two neurons and if there is any evidence of rhythmicity in these two neurons. So how do we answer the question? So any ideas before we actually get to this? What do you guys think? I give you the data, that's all you get. Everything about the neural firing is there in the raster plots. You know what they look like. Well, let me just go back and show you. This is it. I give you this for one neuron and a similar looking plot for another neuron, 76 trials. And that's it. That's all you have. And now your trying to figure out how the brain works, and you're testing certain hypotheses, and you want to check for these two questions. How will you do it?
For synchrony, I think so correlation is the thing that we should look into. So if we have some correlation, that means if we have very high correlation means they are firing in synchrony. If they are
Well,
not,
no,
then
not really. I mean, not really. They could, So the correlation that we studied in the last class, this thing, it was number of spike counts within 250 milliseconds or some such duration, right? So 256 milliseconds. So within 250 milliseconds, you get like 5 spikes, right? They could occur anywhere within that long duration. So just because you have two neurons that are, highly correlated, for example, doesn't really mean that they are firing synchronously, where
uh,
synchrony here is like, millisecond or two. I mean, very occurrence of spike times exactly at the same time.
so then we can have time dependent correlation kind of thing, where
How do
we
you
can
do that?
reduce the time of like you have the like 5 milliseconds interval. Now we can reduce it to 1 millisecond and two, so that we can have this kind of stuff.
So you will do this entire analysis separately on 1 millisecond bins. So,
Yeah, this is one kind of thing, one kind of
okay.
possibility.
Okay. Any other ideas?
I have a much simpler idea. I mean, it's more like, I guess, just a quick code rather than, I don't know whether it would, you would say there's too much of a scientific explanation for it, but for explaining synchronicity at least, I mean, I looked at the raster plot. It looks like a matrix, doesn't it? Right? I mean, with a certain number of rows and columns.
Yeah.
So what you do is you've got ones at the place where you have the spikes. You've got zeros wherever you don't have the spikes. And what you do is you do a simple element-wise multiplication, and then you normalize it by the maximum possible number of overlaps, and that gives you like a certain value as to how much of an overlap there is between two raster plots.
All right. Very nice. Great. So it turns out that's exactly what we do. And this is called a cross correlation function. Okay. So let me explain what this function is. So this is the mathematical definition of a cross correlation function. You so what Srikrut just told us is the value of the cross correlation function at a lag of 0. So suppose I've got two functions f and g. Okay, so these are both time series. So in our neural data, we are just going to look at time series as a bunch of ones and zeros. Okay, so whenever, so I divide my entire duration into 1 millisecond bins. If I see a spike there, I put a 1, otherwise I put a 0. Okay, so, and then I have a similar line for each of the trials for the second neuron. But the cross correlation function so defined is, is valid for any type of function. So let's look at what f and g, two functions like this. So initially what I will do is I will just take a dot product. So this with that, that with that, this with that, and so on, point by point product, and add them up. So suppose I want to look f star g, this is cross correlation at a lag of 0. So n is 0 here. So this is just fm multiplied by gm. For don't worry about the star here. It just means it's a complex conjugate. But we have got real signals, so we don't really worry about this. So at the lag of 0, so f star g 0, I'm just taking a point by point product and summing it up. Just like the way Sweekrut mentioned. So if there are lots of overlaps, lots of ones that are aligned and zeros that are aligned, this sum is going to be large. Okay, and whatever that value is, I'm going to put that value at a lag of 0. Okay, so here, for example, this is your lag of 0. This is how I'm defining it. Here, the two are perfectly overlapping. So the product is large. Now, I shift one of the two trains. This one, as you can see, has been shifted in this, and then I do the same operation, and I put that value, the summed value, at a lag here of -2. Okay, so then I'm shifting this by -1. This is 0. This is shift of 1, shift of 2, and so on. So this function tells you, this is the cross correlation function, about the alignment of the two, the correlation between the two functions here for different values of the time lag. So this is the output. This is the cross correlation of f and g, which is also a function of time, but it accounts for how well the two functions are overlapping when they are shifted by different amounts. This, when I replace g by f, it is then called an autocorrelation function. And notice that when the autocorrelation function, at the lag of 0 is always going to be very large, because the function will obviously be trivially correlated with itself. And as I'm moving away with one function, the the overlap falls away, and then it becomes a triangular shape. Okay, is this fine with everyone? This is the definition of a cross correlation function.
Does it reduce to regular correlation with a lag of 0?
Well, so no, it doesn't. These are two different things. But the regular correlation is defined in terms of the number of spike counts over some interval. And it turns out, if you integrate the cross correlation function over some interval, you'll get a measure that is related to the cross correlation that we defined previously. So there's an integration involved, and the paper that I referred to, Smith and Cohn, they actually show that you can integrate, and that's how the two are related. Okay, and we will maybe touch upon that towards the end of the class today. Okay, so everybody happy with this? So now we are going to look at how cross correlation functions should look like under different conditions. Any questions on this? This is all the maths that we are going to look at. The rest is all based on intuitive feeling and so on. Everybody happy with this definition, right? Pretty soon we will see a variant of this, which is called convolution, in which one of the signals gets flipped essentially. But otherwise, the logic is quite similar. Yeah, somebody had a question?
I was asking, what is the need of shifting the signal? Means like we have.
We will get to it. So let's get to that then. So I used to do all of this on a piece of, on a blackboard in previous years. So I'm going to open a Jamboard now. And I'm going to look at several conditions here when two neurons are firing randomly and so on. And we will see how the cross correlation functions look like in different conditions. Okay. So first, let's assume that two neurons are firing completely randomly. So okay, now, so they're firing in a Poisson-like manner. This is neuron 1. And the neuron 2 is also firing. Whoops, This is going to be a challenge here. So these are the spike trains from another neuron. And this is their Poisson-like. So first tell me, if I plot the autocorrelation of N1, how should it look like? Autocorrelation of N1.
It would spike at 0.
That's right. So when, just try to plot this with a different pen. So this is the same N1 without any lag. So ones get multiplied by ones and 0 get multiplied by 0. So at the lag of 0, there will be a bunch of ones that will be aligned. So it will be a large number there. Now, if I shift the spike train by some amount, what do you think is going to happen? What do I get at some other lag?
Reduced version of it. So if it is like shifted very much, such that there is no overlap, it can reduce to 0.
Okay, will it be 0 or something else? And if it's not 0, what will it depend on?
How much you have shifted it and how much
No,
there
it
is
shouldn't depend on how much I have shifted it. Because they're firing randomly. Your chances of getting a coincident one, a one on both of these trains, shouldn't depend on how much you have shifted it. Because they're firing randomly. The probability of a spike at one location doesn't, really matter.
Aren't they the same spike train?
They are the same spike train. So I'm looking at N1 and N1.
Right.
But I have now shifted N1. It's a lagged version of N1. Sorry, I'm just going to just rub this off. So when N1 and N1 are all aligned, this is the value of the cross correlation at a lag of 0. Then I'm, let's say, looking at the lag of 1. That means every spike has shifted by 1 millisecond. Like this. See, it's a nightmare to deal with this on the Jamboard, but okay. So now what do you think is the dot product, point by point product?
I actually think for small lags it should touch 0, but then as the lag increases a little bit, it'll probably become a slightly larger value, but not the same as what it was initially. That's my intuition.
Well, so it will never reach this level. Here, every spike that you have, well, so the value here is equal to the total number of spikes that you have, because every spike is getting multiplied by itself. So it's 1 plus 1 plus 1 plus 1. So this is the total number of spikes that you have. You will never reach that level. Okay,
Right.
but if they are completely Poisson-like, then actually the lag doesn't matter. The only thing that matters is how many spikes you have in general, because that will determine how many ones you have. So if you shift by a little bit, by chance, how many ones will get aligned? If in this case, for example, in this case, it's actually 0. There's no alignment now between, so there's a 0 here and a 1. So the product is 0. There's a 1 here and a 0 there. 1 here and a 0 there, right? So here it's 0. But if there were a lot of spikes, you would assume that there would be some number of spikes, some number of ones that will still be aligned. And this line would actually depend on, the height of this will depend on the average firing rate, the lambda of the Poisson process. However, if it is completely Poisson-like, then the lag shouldn't matter. The number of coincidences that you might get by chance doesn't depend on how much you shift N1, because it's firing randomly. So it doesn't matter what the probability of getting a 0 or 1 is. It doesn't matter on the lag. It's always the same. You're flipping a coin at each of these bins. And if you shift it, it doesn't change anything. So this is for a random autocorrelation. Okay, and if the neuron 1 was firing lots of spikes, so you had a ton of ones here, still random, still a Poisson process, but with a larger lambda, you would see that this line would shift up. Oops, sorry, this is just, this dot would also go up, but otherwise it would remain flat like this. So this is a Poisson-like behavior. Make sense? So this is autocorrelation. If I talk about cross correlation now.
For even for there is no lag, then also then to it will reach some maximum value, no?
That's right. That's right. So that's that dot here, and it will go to some larger dot value. If I'm looking at cross correlation for a Poisson process, Poisson randomly firing, then it will remain flat, even at 0, right? Because it only depends on what lambda 1 and lambda 2 are, but it doesn't depend on the lag. Okay, everyone happy with this? Say something. It's kind of hard for me to. If there are questions, please ask.
Sir, can you again explain why the cross correlation will not reach the peak and will be constant
Well,
dependent on lambda 1
because
lambda
for a
2?
cross correlation, it's N1 and N2, right? They're both firing randomly. So even at the lag of 0, there won't be any. So this is, so see, look what I have done here. So I am reducing this spike train to a bunch of zeros and ones. So it's a 0 here, 0 here, 0 here, 1 there, 0 here, 0 here, 1, and so on, right? That's now my time series. That's the function that I'm going to use. Okay, and when it's auto, it's the same set of zeros and ones. And then I'm doing a dot product. 0 with 0, 0 with 0, 0 with 0, 1 with 1. So every time there's a 1 here, when there is no lag, it's 1 multiplied by 1, so it's 1. It's basically this thing here. Fm multiplied by Gm at a lag of 0 when n is 0. So, and then I sum them up. So I'm going for an autocorrelation, even if it's Poisson-like, I'm going to get a large value at a lag of 0. Because all the ones are aligned. But if I shift it, then the number of ones that will get aligned will just depend on how many ones are there in the train in the first place. So it will settle at some value which only depends on the average number of spikes, but then it won't depend on the lag because it's firing in a Poisson-like manner. When it's cross, there is no alignment between N1 and N2 to begin with, because they are both firing randomly. So the cross correlation function will remain flat and will only depend on the total number of spikes in the N1 train and N2 train, but not their position relative to each other.
Yes, and thank you.
Okay, everyone happy?
So
Yeah.
I had a small query, and this might be slightly, it's just slightly counterintuitive to me. So in even in the raster plot, there is an upper limit on the number of signals you can see in the interval you're measuring, right?
Yes.
Like the number of spikes. So if you have two such processes, then there has to be an upper limit on the on the sum or the number of ones that coincide.
That's right. So that's this upper limit here, T.
No, but in case for cross also,
Uh-huh.
this should be, it should theoretically take values right from 0 till that upper limit, right?
That's right. So this value can't go too high. You're right. So this will actually be a lower number, finite number that will just depend on lambda 1 and lambda 2.
Okay. Yes,
Okay,
sir.
and another thing that you now that you pointed out, I should mention that this cross correlation function will remain flat only when the spike train is infinitely long, because no matter how much you shift, you end up the same number of coincidences. But let's say you are only looking at 200 milliseconds. Okay, so then you've got two spike trains that are only 200 milliseconds. Obviously, if you shift something by, let's say, 100 milliseconds or 150 milliseconds, so the second one now becomes something like this. Like this. So the potential overlap can only occur over a very small interval because it's finite here. It's 200. And obviously, if I move it by more than 200, then there can't be any overlap. So for finite length signals, even if they are Poisson-like, you would expect that the correlation function should have some sort of a shape like this, just because after this, there are no ones and zeros that are sort of coinciding. Right? So this sort of a function is true only for an infinitely long signal.
Yes, sir.
For
That's what I wanted
finite duration, we have to account for the fact that the area of overlap is reducing. Okay, and we will see both of that in the coming slide. Okay, so everybody happy with random now? This was random. So I'm going to now introduce some more things so you will appreciate why the cross correlation function is so useful. So now tell me, now let's look at the second one, delay. I've got one neuron which is connected to another neuron. So whenever the first neuron fires, the second one fires after 5 milliseconds. So my neuron 1 is firing in a Poisson-like manner, and neuron 2 is just following it. What do you guys expect the cross correlation and autocorrelation to look like? What do you guys think?
For cross correlation, if the lag is exactly 5 milliseconds, then it would be very high.
That's right. So this is the answer to your question, that why do you want to look at lags? So here your auto will look like before, this assuming this is a Poisson-like neuron. So auto will be like this for both the neurons because they are just shifted, and the cross would have a peak at 5 milliseconds. So you can actually look at delays between neurons by looking at the cross correlation function. If you shift this by 5 milliseconds, then all the ones are aligned and you get a large value. Correct? Okay, I'm assuming everyone is happy. So,
guess about the, is it related to rhythmicity also, no? Because
it's
we
what?
have, is it can be related to rhythmicity?
We'll get to it. Yeah. Right now, there's no rhythmicity here. It's just one neuron firing in relationship to another, but they are both Poisson-like. This is completely random, and so is this. They just have an interrelationship between them. If there is refractory period, what happens? refractory.
So why would a refractory period have any effect on the cross correlation?
What about autocorrelation?
No, right? The refractory period will only affect the minimum time interval that can occur between two spikes.
So
It should
how
not
does
affect.
that? How is it reflected in the autocorrelation?
So I think if the time lag between like the time, okay, there's no time lag in, yeah, the time lag between the spiking within the neuron is less than the refractory period. In that case, the autocorrelation may be quite small.
Very good. So it will actually change like this. Oops. Just because, right? So you've got a spike train, it's firing in a Poisson-like manner. Poisson-like now, not completely Poisson. But if I shift it by one, a very small amount, there's no chance of having another spike. You will never have two spikes that are close to each other because of refractoriness. So there is going to be a dip because if I shift my neuron spike train by 1 millisecond or so, and the refractory period, let's say, is 5 milliseconds, then there's absolutely no way that you will get and have another spike there. So it's going to look like this. This is exactly what someone mentioned, right? So you can actually understand a lot about the behavior of the spiking activity by looking at just the cross correlation function. Let's look at oscillations. So oscillations now. Man, this is very hard to do this with a mouse. Oscillations. Suppose a neuron tends to fire rhythmically like so every 25 milliseconds or so. So this is 25 milliseconds. What is the autocorrelation function going to look like? What do you guys think?
We would have various spikes at the interval of 25 milliseconds.
That's right.
It has a
Very easy, right? So it's spiking at 0. This is the lag. At 0, of course, everything is aligned. You shift it by 1, it falls to 0. Shift it by 2, it falls to 0. And it will remain 0 until you shift it by 25 milliseconds. Because at that time, this spike would have moved to the next one. This one would have gone there. The whole thing is shifted now. So then you will again see an increase. And again at 50. So it will, so if there is some noise here, it will actually start looking like this. Okay, so this is the autocorrelation function of a spike train that seems to be firing rhythmically, and some noise would if there is no noise, then it will be peaky like this, just these delta functions, just these sharp values. If there is some noise, then it will look like this.
Excuse me, sir.
Yeah.
Sir, if we do this autocorrelation, then at 25, we will lost two spikes. Na? So uh, the magnitude will decrease
So
very
you're
small.
saying we would have lost this one
Yes,
and
and the last
that
one.
one. Yeah. So it will decrease. This is what I had mentioned here also. here also, for finite signals, you have to account for the fact that the overlap is reducing. What I'm mentioning here is for infinite signals, infinitely long signals.
Okay,
If it is finite, then the whole function will get multiplied by a triangular function, which is the reduction in the overlap between the finite regions. So you are right. But what I'm mentioning here is for infinitely long signals.
okay, sir. Thank you.
Yeah. Okay, so oscillations. When there is synchrony between two neurons, you would expect, so this is this rhythmicity you see in the autocorrelation itself. If two neurons are firing synchronously, then obviously you will see a spike at a lag of 0. This is the special case where the delay is 0, right? So then the cross correlation function will show a spike at 0, showing that the two neurons are firing in synchrony. Okay, so I won't make that again. Last one, if they are bursty, what does that mean? Bursty means that spikes tend to occur in bunches like this, a bunch of spikes, and then it's silent. Then again a bunch of spikes, then it's silent. Again a bunch of spikes. Whenever spikes occur, they occur in bunches. There are neurons that do this kind of behavior. So for in this case, the autocorrelation function has actually a sort of bump like this. This is the opposite of refractoriness, because they are bursty. Even when you shift it by a little bit, let's say 1 unit, there's still a fair bit of overlap that you see here. Okay, so for small lags, there's higher overlap, and then it becomes sort of random. So you can learn a lot about how neurons fire, even an individual neuron, as well as how neurons fire relative to each other by looking at the cross correlation function. And it's such a simple function, the MATLAB will just, get it with a single command. And it conveys a lot of information about how neurons are. So now we are ready to look at what happens in our real data. Any questions on this? Otherwise, we'll now move to the data and answer the questions that we asked.
For bursty, so there you show only one peak. There may be multiple peaks such like that, no?
No, why should there be multiple peaks?
Suppose you have these burstness, and then you said that there would be some interval, and then it will have this burstiness again.
Yes,
so,
but this interval is random. You're talking about a condition when there is burstiness and rhythmicity.
Okay.
If that is the case, then you'll have a second peak. But right now, the bursts themselves are random. So there's no dependence. So yes, if they are bursty and rhythmic, then you will have multiple peaks. But the other peaks are showing rhythmicity, and the burstiness is reflected in this sort of a shape. Okay. All right. So, okay, so this is what we do then. I put a 1 when there is a action potential and a 0 otherwise. And then there are two things I have to do. One is I need to account for the fact that just the number of action potentials might be different. So I normalize it by the mean firing rate, such that just because the absolute firing rates are high doesn't sort of change the metric. So this is called a cross correlation function, and in this case, it's a normalized cross correlation function which accounts for the number of the fraction of coincident spikes. It's normalized by the absolute firing rate. In this paper, they discuss why they use this form of normalization. Other forms also, but here we stick to this. And this is the other factor that accounts the finiteness. So this t minus tau is a triangular function. When tau is more than t, this is 0. So otherwise, when lag is 0, you are dividing by a bigger number, because that's there you have maximum overlap. And as the overlap reduces, this denominator becomes smaller. So this accounts for the fact that in a finite signal, the overlap changes as the lag increases. Overlap reduces as the lag increases. So you penalize the smaller lags more than the larger lags. Okay. So this is the function that I've given you a code that implements this. So if you run it, you will get to the data. Before that, I would just sort of tell you what to expect if the binding by synchrony hypothesis is true, which means that the neurons are firing rhythmically. Then the autocorrelation functions ought to have this sort of a shape. Okay, so this is evidence of oscillations, this rhythmicity at 25 milliseconds. We have talked about this. And actually, the real data, this is about 2 decades ago, 3 decades ago now, showed that indeed neurons tend to fire rhythmically. And if you compute the cross correlation between two neurons, it shows this rhythmicity as well as peak at 0. So this is evidence of synchrony in a rhythmic manner. Neural assemblies fire synchronously at gamma frequencies, but only when the stimulus that is presented is some unified whole, not broken into pieces. So this is the study that sort of made this whole thing very popular. They showed a long bar to on the receptive field of two neurons, and when you move this, the two neurons fire rhythmically as shown here and also synchronously. But if you introduce a small cut in the bar so that they are two different objects now, even though the individual neurons are still seeing the same thing and they are firing sort of rhythmically to the small pieces, the synchrony reduces from 0.3 to 0.2 and is abolished when the two bars move independently. So the grand idea was here was that when you look at a unified object whose attributes need to be bound together, this binding is achieved by synchrony at gamma frequencies. So gamma oscillations induce synchronization that somehow cause all the different small fractions that individual neurons see. This binding occurs, and then you see the full picture. This is this grand idea that when I look at an object, different neurons are seeing small parts of it, but I have this feeling that it's one unified thing. So who is doing this binding? The binding is done through gamma synchronization in their data. Okay, that is actually why gamma oscillations are very popular in the field. It's something that's related to these high level mechanisms related to oscillatory synchrony. Okay, and my lab actually studies this, so we spend a lot of time doing all of this, and we will see in our data evidence of gamma synchronization as we go along. Okay, in the same data set that you have with you. So let's look at what the auto and cross correlation functions look like. So let's look at auto correlation first in our data. So I'm looking at this 250 millisecond interval, and this get CCG is the function that does this, and this is the output. Okay, so the number of coincidences is flat. At 0, of course, it spikes to 1, becomes a very large value, and then otherwise it's 0. So this again is, well, at 1 you see a bit of a dip here. It's a very small dip, but otherwise it remains flat. So this is evidence that the neuron is firing in a Poisson-like manner. Okay, is that fine?
So
Now
the dip,
let me ask the question.
the dip is because of the refractive period, right?
That's right, yes.
Yeah.
So now let me ask a question that, hey, in the initial period, 0 to 256 milliseconds, is the neuron still firing randomly? And if I do this, I get this plot. Now you say, this looks odd. The neuron was not firing in a random like manner. You see this evidence of this burstiness and a sharp refractory period, and then it flattens out. But turns out this is an artifact. When we do real analysis, real data analysis, you have to be aware of the fact that there could be all sorts of artifacts. Now, what kind of artifacts are we talking about? Note that, I guess I have to go back to the present, to my, to here. So note that I'm looking at N1, and well, so it's this autocorrelation again. So note that there are a bunch of spikes at one point and then it reduces. This is as a function of time, right? That's what you see here. Initially the firing rate is high and then it becomes lower. And because of this, if I shift this spike train by a little bit, I have a larger chance of having coincidences, because the spikes tend to be bunched up at one place and then it is thinning out at a different place. But this is true even for a different trial. If I look at any other trial, I will still see. So here, look at this. I have got trial 1, so bunch of spikes together. This is trial 1. Trial 2, again a bunch of spikes together and then fewer spikes later. Every time this is happening. This is neuron 1. And again, let's say I look at the same spike train here, number of coincidences. So I am interested in what is the fraction of time when the neuron is firing at the same time as the other neuron. Okay, if it's N2, that's probably easier to understand. So I want to look at coincident firing, the firing timing of this and this being at the same time. But just because the average firing rates are higher and then they reduce, the average firing rate is like this, I'm expected to get more coincidences, more number of ones aligned together at one time, just because they seem to be bunched up at this time. And this is not evidence of synchrony. This is just the fact that the firing rates seem to be co-varying because they are related to the onset of the stimulus. I need to account for this. The way I account for this is by looking at the correlation of this spike train with the next one here. Okay. If I look at the correlation of this spike train with the spike train of neuron 2 for a different trial, so they are not being recorded simultaneously. This is a little bit involved, so please stay with me here. So this is the spike train from neuron 2 for a different trial. And if you see any coincidences between this and this is just because of covariations in firing rate, not because of synchronicity related to them firing at the same time. So this is the average correlation, the spurious correlation that you expect to see just because of slow fluctuations in firing rates. And you need to subtract that out. Okay, this is a tricky concept, so you'll have to really spend time on this. I have given you an assignment. So this is there in Baer et al. 2001. This is a homework assignment. There are different ways of doing what is called a shuffle correlation, which is essentially the cross correlation of non-simultaneous spiking. You can only do this if you have got multiple repeats, which we do. We have 76 repeats. So you can look at the cross correlation between non-simultaneous pairs. And why just do one step cross correlation as what you are doing here? You could actually do a cross correlation of all possible cross pairs which are not simultaneous, which is called an all-way shuffle correlation, which is given by this formula. And this is your homework assignment. You have to figure out where this formula comes from. All the information is given here in this paper and Smith and Cohn that we are referring to. This is not straightforward. So I'm going to leave you with this. This is one of the homework assignments for you. But if you do the shuffle correlation, you will find that this shuffle correlation looks like this cyan train. So once you subtract it, you actually get back your original one. So it is still in a Poisson-like firing with a little bit of refractoriness. The small bump that you see here was just because that the firing rate was changing and that was causing some spurious correlation. And once you account for that, you see that it is firing in a random Poisson-like manner. And this is the autocorrelation. If you look at the cross correlation, it remains flat without the without any correlation. And even in the early, this 0 to 250 millisecond, even though there seems to be some synchrony, the shuffle correlation shows the same thing and the overall correlation remains flat. So there is no evidence of synchrony either. So in our spike train, there's no evidence of rhythmicity and there's no evidence of synchrony. And in the next part of the module, I'll show you that both of these conclusions are actually wrong. There is actually evidence of gamma synchronization in this data set. It's just very hard to see. Okay, I've run out of time, so I will stop here. So please, I will give you the assignment today. Please start working on the assignment. It's due in about 2 weeks. We won't be able to discuss this paper in a journal club like fashion. I will going to briefly summarize it in the next class, and then we will start with the local field potentials. So our first module comes to an end now. I'm just going to summarize this in the next class and then we'll be done. Okay, so module 1 is over and I'll stop recording now and I'll take questions. Anyone who